Well, hello and welcome. Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Peptide Aggregation in Manufacturing Mechanisms, Challenges, and Case Studies. My name is Soumya Shashikumar, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, and that can be found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I would also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I would like to thank Polypeptide Group who developed the content for this presentation. Polypeptide Group is a specialized contract development and manufacturing organization, or CDMO, for peptide and oligonucleotide-based active pharmaceutical ingredients. By supporting its customers mainly in pharma and biotech, it contributes to the health of millions of patients across the world. Polypeptide serves as a fast-growing market, offering products and services from preclinical through to commercial stages. Its broad portfolio reflects opportunities in drug therapies across multiple areas with significant focus on metabolic diseases, including GLP-1. And now I would like to introduce our speakers for our event. Dr. Rekai is the head of process and analytical development at the Polypeptide Braine-l'Alleud site in Belgium. She holds a PhD in organic chemistry with a focus on sugar chemistry and completed postdoctoral work in polymer chemistry. Dr. Rekai began her career in January of 2000 as a peptide R&D chemist at UCB Bioproducts, later specializing in chemical upscaling and process validation. At Polypeptide, formerly Lonza and UCB Bioproducts, she oversees development and manufacturing processes across all clinical phases, from R&D through to commercial production. And Dr. Anais Pouzol-Collinson is a downstream process scientist at the Polypeptide Braine-l'Alleud site in Belgium. She brings over a decade of experience in manufacturing and R&D. Dr. Pouzol-Collinson is an expert in peptide and aggregation analysis, particularly amyloid-type peptides. She has built and led biophysics teams, applying techniques like TLS, FTIR, SEC, CD, and fluorescence to advance peptide categorization. She has contributed significantly to structure activity understanding and to the development of biologic formulations in preclinical studies. And Dinesh Parmar is the head of regulatory affairs at Polypeptide's Ambernath site in India. With over 16 years of experience in API regulatory affairs, he has filed more than 50 US FDA Drug Master files and has worked on critical responses from major health authorities, including the US, EU, Canada, China, and South Korea. Dinesh has strong experience in the realm of early-stage product development and product lifecycle management and forming regulatory strategy. His approach helps streamline approvals, align with customer needs, and support timely commercialization. And now, without further ado, I would like to hand the mic over to our first speaker, Dr. Rekai. You may begin when ready.
Thank you, Soumya, for the nice introduction. All right, so ladies and gentlemen, good morning, good afternoon. Welcome to our webinar, Peptide Aggregation in Manufacturing, aiming to share with you a crucial topic in the field of the biotechnology and pharmaceutical manufacturing. We know that peptide aggregation is really a complex phenomenon that can significantly impact the quality and the efficacy of the pharmaceutical product. So therefore, understanding the peptide aggregation challenge is essential for developing the right strategies during the manufacturing process. Today, we are pleased with my colleagues Anaïs Pujol and Dinesh Parmar to share with you how we have overcome some aggregation challenge at Polypeptide. So to start with, a short introduction on the peptide aggregation phenomenon, followed by the fundamental, the mechanism that drive that process. Then we will move to a selection of analytical tools for detection and characterization of aggregates. We will then highlight some challenges in the peptide manufacturing with illustration with case studies, and finally, some regulatory considerations that are essential for a successful product approval. So let's start by a brief introduction of the peptide aggregation in the drug substance and the drug product. So peptide therapeutics market is really growing rapidly, and due to the need to treat some chronic metabolic and infectious diseases. It's important to highlight that peptide aggregation will play a crucial role in the biological processes and in the disease. In the case of the functional aggregates, the peptide aggregation will be a normal and a necessary process. This is the case for the bacterial biofilms, as well as for the hormone storage. We have hormones that are stored in aggregated form within secretory granules in cells, like the insulin. But then the peptide aggregation starts to be a challenge when we are addressing neurodegenerative diseases, where we have some misfolded peptide that aggregate into amyloid fibrils that accumulate in the brain and that will disrupt the cell's function and lead to the cell's death. This is also the same for the type 2 diabetes GLP, where the peptide hormone will aggregate in the pancreas and therefore forming amyloid deposit that can be toxic to the insulin-producing cells. It's important to highlight that the type of aggregates will depend strongly on the amino acid sequence, and that the peptides, which are smaller than the protein, will fall in only one type of structured or unstructured aggregates. So considerable challenges are faced by the peptide drug substance and drug product manufacturer with respect to the development, production, and formulation of those peptides due to the physical stability of peptide that can generate either amorphous aggregate or some structured fibrillary species. If we look to the publication of the Royal Society, edited in 2017, we can see a series of very well-known peptides and with fragments of GLP with their respective length, and we can see that all of them will generate some fibril aggregates in a different condition of pH. pH could be neutral pH, acidic pH, or basic pH. So the peptide aggregation can occur in various forms. We can have some subvisible, gel, or precipitate. And when aggregation is occurring, then we have big challenges in matter of functionality, as aggregated peptides lose their intended biological function, compromising their efficacy. Aggregation can also impact the toxicity of the cells and lead them to some cell death or some dysfunction, or trigger as well some immunogenicity responses that may lead to adverse reaction in patients. That's the reason why a lot of effort has been devoted to explore a new chemical modification in order to improve that stability and to inhibit initiation of aggregation. And this is the case for the GLP-1 analog, which are used in the treatment of type 2 diabetes, very well known to be prone to aggregation. So let's now explore the fundamentals and the mechanism of the peptide aggregation. So the peptide aggregation is a process where individual peptide molecules will come together to generate larger and soluble structure, starting from the monomer up to the fibrils, passing through the low and the high molecular oligomer, then the photofibrils. We know that that phenomenon is significant in various biological and medical diseases, and that aggregation process can lead to formation of various Among those structures, we can have some amorphous aggregation, where we have a non-structured irregular aggregates, or we can have the fibrillar aggregation, where we are generating, in that case, some structure known as the fibrils, and those fibrillar aggregates are often associated to the amyloid disease. We can have oligomeric aggregation, where we are generating small and soluble aggregates called oligomer, which are oftentimes intermediate to generate the larger aggregates. And we have also the crystalline aggregation, where aggregates are highly ordered, crystalline structure, and usually those aggregates are much more stable and less prone to causing toxicity. For a successful folding, the process must be energetically favorable to get the thermodynamic stability. And nowadays, we have the computational chemistry that can help to predict the folding. A series of algorithms as the Rosetta, Tango, and the Zigregator as example. So now let's move to the three key stages leading to the peptide aggregation. So the first one will be the lag phase, where we have a stable nucleus that is generated. So during that period, as you can see on the graph on your left, there is little to a no visible aggregation, as monomer and small oligomer will interact to generate the prefibrillar structure. Then start the exponential phase, where we are getting larger structure. And during that phase, monomer and oligomer will be added to the nucleus to generate those larger structures. After that, we have the first stage, which is the plateau phase, equilibrium, where the aggregation process will slow down. And in that case, we will have the right balance between the new aggregate generation and the aggregate dissolution. But what is very interesting as well to see is a seeding effect in the aggregation. So if we add some seeds into the solution of the monomer, we can shorten significantly the lag phase. If we have low seeding, we can have a half of the lag phase short term. Whether if we have a very high seeding, then we have almost no lag phase occurring. We have directly the exponential phase and the rapid growth of aggregates into solution. So the peptide aggregation, as already introduced, is a very complex phenomenon. That's the reason why extensive research programs have been undertaken in order to increase the understanding of all the mechanisms and the different factors that can influence aggregation initiation. So here is a combination, so it's 4 cases with a combination of different factors that can lead to aggregation. In the first case, we have the solvent, the seeds, and the ionic strength. In the second case, we have a counterion, concentration, and impurity. Third one will be the shear stress, temperature, and oxidation. And for the last one, it can be the transportation, filtration, and storage. Already through those 4 cases, you can see that several factors could be involved in aggregation, and a combination of those parameters will initiate the aggregation. So let's see now on the next slide the different factors that can be at the origin of the aggregation. The first one will be the intrinsic factors, which are inherent to the peptide itself. So it will be, of course, the amino acid sequence, the net charge, the hydrophobicity, the chemical degradation, chemical modification, or any related impurity. Then we have the second part, the extrinsic factor, which are linked to the external conditions. So in that case, it will be the excipient with the buffer, the salt, organic solvent, and surfactant. We have the concentration, pH, temperature, the pressure, ionic strength, type of agitation with the stirring or the shaking mode, the foaming, the pumping, or external impurity like the metal, extractable, leachable, and silicone, the surface adsorption, the interface air between the air and liquid, creating some hydrophobic interfaces, and of course the operational time. So you can see that this is very complex because aggregation can involve a series of factors, and a combination of them will lead to that peptide aggregation. So now I will pass I will hand over to Anaïs, who will introduce the selection of the analytical tools for the characterization of the aggregates.
Thank you, Joa. So we're going to have a look at which analytical tools can be used for the detection of aggregation. So when we look at an aggregate, we need to consider several factors. The first factor will be the particle size of the aggregate, as they can come in different sizes. A small oligomer won't have the same size as a mature fibrils. We will look at the structure and the physical arrangement, the physical property of the surface of the aggregate as well. If they're hydrophobic, hydrophilic, which kind of secondary structure are they arranged into? And then it will be the shape and the morphology of the aggregate. So according to what we want to see, we will select a different type of technique. Not all the technique will give the same information, and quite often we will have several techniques to understanding of the aggregate. So in terms of looking at the particle size, the techniques mostly used is dynamic light scattering, the DLS. Is a good technique to identify small particle versus large particle. It will so it scatters the light according to the size of particle. Small particle will scatter the light faster than the large particle. The downside is that well, the fibril is not spherical, so you have effectively some issue with the quantification of the size. So it will mostly use for qualitative purpose for when you work with amyloid fibrils. However, it's a really good high throughput technique to quickly identify if there is big particle in a sample. One of the last technique is a laser diffraction for particle. This one uses a different type of scattering. You use the mis-scattering theory. Is a good quantitative technique. For this one, you have an absolute numbers of the particle size. One as well of the last one is a nanoparticle tracking analysis. Again, is more sensitive than the DLS. You have a good size distribution and a good particle size measurement. Is in liquid is dynamic. So when you measure, you can still record it, what's happening in your sample. And then you have the background membrane imaging, which is quite similar to the flow chem or flow imaging technique. However, it has better improvement in a way that it's a high throughput technique. You put your sample as a drop, and then you vacuum fluid liquid, and your particles stay on the membrane, and you analyze this membrane. If you look at the molecular weight of your aggregate, you will use a size exclusion or ultracentrifugation technique. The size exclusion in general is coupled with a mass detector. The disadvantage of the size exclusion with a peptide is that sometimes it's difficult to find the right column to be able to separate your monomer to your aggregates. Also, it would be better to use this technique on a low pressure system instead of high pressure system. And for the analytical centrifugation, you will have the same issue if you aggregate are too close in a molecular way or hydrodynamic radius. You will have difficulty to separate them by this technique. Then we go to a technique which gonna look more in secondary structure or physical property of the aggregate. The Fourier transform infrared is a really good technique for secondary structure analysis. The advantage is that you can analyze your sample in solution and in a solid state, so on your final API. When you are using solids, you can do some kinetic sample to see the evolution of your sample over time. The disadvantage is that you can have a strong matrix interference with this technique. However, the measurements are pretty fast with this technique. The circular dichroism is as well another really good technique for secondary structure. Again, it has some matrix interference, so you will have to see which type of matrix will fit best with this technique. It's a good technique because it uses low concentration. However, it's a big instrument. It's not always easy found in company, but so it may require university collaboration. Then you have the fluorescence dye. So the advantage of the dye is that they're going to recognize a specific region of your aggregate, a specific arrangement. For instance, the one with the most famous is the THT dye. It will recognize a specific arrangement of your beta sheet. However, the THT recognize mainly mature and protofibrils, so not the early aggregates as oligomers. So you may want to use different type of dyes as well to characterize your aggregate, depending on if you want to look at the smaller size or the bigger size. But it's a good technique to use. You have as well some available computational technique who can fit the THT kinetic if you do a kinetic and quantify properly the fibril in the sample. Finally, you have the EEC chromatography, which is a complementary technique to the size exclusion in a way that it will look at the hydrophobicity of your aggregates. And if your aggregates have a different type of hydrophobicity that your monomers, then you will be able to separate them and detect them and quantify them. And then the last technique, and not the least, is the one who allows to see the shape and the morphology of the aggregate. So they are microscopy technique. So you have the transmission electron microscopy and the atomic force microscopy. So they both really good technique. You can properly see the oligomers, protofibril, and amyloid fibrils. You can see some examples. This is the IFM technique, but you see the same on the time using the time technique. Really, the oligomers are really small compared to the fibril when you can really see long hair structure. With the IFM, you can even go into detail if you are interested to see if your fibril twists, or you can as well use the IFM to properly measure the length and the thickness of your fibril, which will be more difficult on the TEM because the pictures are less sensitive. But you can, on the IFM, you can really quantify. So you can do kinetic measurement with both technique. The sample preparation is not too invasive, so you won't modify too much your aggregate. That's quite often what people are scared using analytical. Will I modify my aggregates with microscopy? Is quite safe. Also, the IFM is a small technique that can easily fit on a bench, which is not the case for the TEM, where you will again need university collaboration for that. So I pass again the next slide to Joa.
Thank you. So now we will dive to the peptide aggregation challenge that a peptide, drug substance or drug manufacturer can face by sharing with you 3 real case studies that we have faced at polypeptide and how we have overcome them. So first of all, to produce a peptide, we will need to perform the upstream part of the process that will consist to build the protected peptide, either by a liquid phase peptide synthesis or a solid phase peptide synthesis. Then after getting the protected peptide, it is submitted to a cleavage in acidic condition to get the crude peptide, which contains the targeted peptide and all the related impurities. Usually, during this upstream part of the process, we have a very low risk for aggregation. But as soon as we are starting the downstream part of the process, over the 6 process steps that you can see on the slide, the crude extraction, the purification, if we have a primary and secondary, the salt exchange step, evaporation, microfiltration, lyophilization, up to the final isolated API, we have a very high risk for aggregation initiation. So let's share the first case study, which is an historical peptide having a structure of more than 35 amino acids and belong to an amylin product. In that experience, we have seen a significant gel formation during the downstream processing at the step of the purification, salt exchange, and evaporation. Our solution, which was a clear solution, turned slowly into a hazy solution. So then we could demonstrate that the physical change was linked to a physical conformation change linked to the amide bond. So the CO stressing vibration is very sensitive to conformation. And in that case, we could see the wavelength of the alpha helix that is at 1652 moving to the 1630. So the aggregated peptide contain mainly the beta sheet motif as secondary structure, in contrast to the alpha helix for the API, which is a native predominant secondary structure. So we have then studied the evolution of our peptide in solution over time, and we could see through the FTIR second order spectrum the increase of the amide bond 1 at 1630, highlighting this conformation change with a very strong increase of the beta sheet motif into our peptide. In order to understand the like phase of our peptide, we have taken 6 samples of different batches that has been produced in the same way during our manufacturing process, and we have studied the like phase. And we can see that from the 6 samples that were solubilized at a concentration of 15 milligrams per milliliter in 2% of acetic acid, that some samples have a like phase quite close to 5 days, where others had a like phase of 8 days. But all of them, when aggregation was starting, then the kinetics increased strongly. So in order to understand the parameters that were at the origin of the aggregation initiation, we have performed some screening study in the laboratory through DOE approach. And among all the parameters that has been studied, 4 of them were highlighted as impacting the gelling kinetics. The first one was the peptide concentration, a too high peptide concentration that needed to be reduced, the pH range, the process duration through our operation step that also need to be reduced, and all the foaming during the stirring step. So by addressing those 4 parameters, we were able then to have batches that could be produced with no anymore initiation of aggregation. The condition needed to be changed. So then the second case study is a peptide, also a long one, more than 30 amino acids that is belong to the GLP family. In that case, the aggregation has occurred at the level of the crude extraction. So we have seen some visual solution change from a clear to hazy solution that you can see also on the picture when we were starting the filtration through our different cartridge. And again, through the FTIR technology, we were able to see the conformation change mainly to beta sheet motif as a secondary structure. So therefore, we have performed a root cause investigation on the laboratory by selecting a series of parameters that we were believing as to be the parameter affecting the aggregation of initiation. So the buffer composition, the stirring, the temperature, and the pH. We have performed 3 DOE. So the first one was a pH and temperature variation. The second one was a stirring step variation. And the third one, the buffer composition variation. What was very interesting to see is that not only one parameter can be at the origin, but also when we combine some parameters, they can also have an aggregation effect. So the high temperature combined to the low pH also influence the haziness of the solution. In that case, we have also the buffer composition that was also playing a contributing role. Then we have a try also to initiate those aggregation through some elemental impurity, but it was not the case, not a parameter influencing the aggregation, and then the process duration. So through the process, we have different steps. And when we look to the process duration at the laboratory, we could have also aggregation linked to that particular, to that factor. So the last case study is a shorter peptide compared to the preceding peptide. It's a non-GLP family peptide. In that case, we have a visual solution change from a clear to an easy solution during the homogenization of the purified fraction just before starting the evaporation steps. So we know that in that case, that the steering, mechanical steering was a key contributor to the initiation of aggregation. Therefore, we have studied in the laboratory the propensity of this peptide C to turn into a complete aggregate state. And what we could figure out is that the solution with no steering after 22 hours remain completely clear. But if we let this solution in a continuous steering, overnight, we have an aggregation, a peptide completely, a solution completely aggregated. So, meaning that the mechanical stirrer is a key parameter in the initiation of aggregation. So, in order to understand also how the different batches were behaving from a mechanical stirrer, we have conducted in the laboratory some aggregation kinetics lab study under the sporadic steering. So, and we could see that depending on the batch that we have produced, different have a start of lag phase. 4 of them were quite short lag phase, but the batch number 3 has a much more longer lag phase. So in order to understand what was different from the different batches, we have screened the different master batch record and our different parameters. We were able to see that the mechanical steering was confirmed, so the batch number 3 was still shorter, and the speed was also different. So we have tried to fine-tune that parameter, but whatever we were doing, we have still just a delay of initiation of aggregation and not an erase of a complete aggregation. So therefore, what we needed to do to overcome completely the aggregation was really to change the evaporation matrix at the level of the purification salt exchange by changing the solvent and having the solvent where the peptide could be much more soluble. We were able to completely erase this aggregation initiation, and even after 20 days, we do not have any aggregates occurring. So now I would like to hand over to Dinesh for the regulatory consideration.
Thank you, Johar and Anis, for bringing aspect on chemistry manufacturing associated with peptide aggregation and describing different methods associated with to study peptide aggregation. If we see from the regulatory point of view and health authority point of view, peptide aggregation can bring physical instability in formulation, and it can raise the safety concern and can bring immunogenicity concerns. So it becomes very important for us to study the peptide aggregation from the regulatory perspective. Whereas if we see from the industry and manufacturer point of view, peptide aggregation is a complex topic as we have described, and it can be influenced by several factors, intrinsic and extrinsic factor, and there can be different size, shape, and morphology of peptide aggregation. Unfortunately, there is no single method available which can screen the entire spectrum of the peptide, and that's why each agency or the guideline also suggests that we need to employ a different orthogonal tool. So considering all this aspect, having robust regulatory strategy and systematic approach to bring consensus with health authority is important. Yeah, before taking into the actual regulatory consideration, maybe the next slide, please. I would like to take you through different terminologies and the guideline associated with on this topic. So these are the different terminologies which are around the peptide aggregation, and oligomer is nothing but multimers formed from the monomer molecules, and this can be a covalent and non-covalent multimer both. Whereas aggregates are something which are always a non-covalent multimer, and these are formed due to the weak interaction like hydrogen bonding, van der Waals forces, and these are very sensitive. That's why during analysis we need to provide a favorable and native condition to preserve that aggregates to have right quantification on this aspect. And then high molecular weight impurities are something very broad terminology which includes covalent and non-covalent multimer. And covalent oligomers are nothing but a oligomer, as name itself indicates. These are permanent, and this are formed due to the disulfide bond, and this can be detected by SEC method. And non-covalent aggregates are nothing but the aggregates formed from the weak interaction. If we see the guideline expectations, there are different guidelines available and which describes the expectation on peptide aggregation. So the first one is USP General Chapter 1503 describes that we need to study the structure conformation changes and peptide polymerization and employ a different analytical method. So when we study the protein folding and protein misfolding, so similarly the peptide will also have a secondary and tertiary structure and will have a conformational structure. So due to this intrinsic or extrinsic factor, this may take unconventional path and form aggregation. And as we explained, there could be a different type of aggregation need to be studied with different analytical methods. If you see the FDA rDNA versus synthetic peptide guideline on generics, it also says that we need to demonstrate that peptide does not increase the aggregation propensity, and especially under the stress condition, we need to study the nature of peptide aggregation. And lastly, the similarly EMA draft guideline on development and manufacture of synthetic peptide also describes the same thing, that aggregation may occur in the synthetic peptide and could be potentially related to safety issues. And we need to study the nature of peptide aggregation. And one more layer they have gone down and they specified that fibril aggregation need to be studied with the TST assay. Next slide, please. So this is the slide where we are going to discuss a regulatory consideration describing the different aspect. On the most left hand side, you will have different activities and some recommended analytical tools, and DMF section where this information will be included, and responsibility of API and formulator. So we typically start with characterization, and as I mentioned, as the length of peptide increases, the chances of having secondary tertiary structure or higher order structure is high. So we need to study a desired oligomeric state of the peptide, and that can be studied with the CD, circular dicrosium, NMR, DLS, and SVAUC method. And this information typically included in characterization section 32S31. And I believe this goes with the responsibility of API and formulator both, because formulator would like to study the oligomeric state in the solution condition of the diluent which they have. Once this is studied, we believe that performing a risk assessment would be very important. And typically we can see during manufacturing, if apparently if subvisible, visible, or insoluble aggregation, gelation, or precipitation happens, and that can be simply studied with optical density or turbidometry method. And this this information will go in 32S26 section or development section and belongs to API manufacturer. And if such issues are there in the peptide process or during manufacturing, then it would be important to identify the critical parameter and the potential parameter influencing aggregation. And this information would typically be presented in 32S22 manufacturing section and 32S26 development section. And if we see that the peptide aggregation is persistent in the molecule, then we need to have a control strategy. And a control strategy can be established with if you have fibrillary aggregation, then a TST method is good. Otherwise, you can have FTIR, optical density, or DLS per se. And when we go for the API-based studies, when API is manufactured in the solid form, we typically get question from the agencies to study covalent multimer and as and discussed, SEC method is sufficient enough to study the covalent multimer. But when it comes to non-covalent aggregates, which are as I mentioned, these are very sensitive, and we need to provide a favorable condition to preserve that aggregation. So a native method or non-organic method need to be developed, and there we believe that SECMALA's multi-angle light scattering, TST or DLS could be helpful. And this particular information will go in the impurity section 32S32, and if needed, we need if the peptide aggregation is persistent, then specification control would be needed, and that should be part of 32S41 section. And lastly, we believe that when API solution stability need to be studied in the formulation condition, that belongs to a formulator, because they are more aware of the formulation vehicle and the diluent and the excipient which will come in the contact with the peptide. So the similar tools like CD, DLS, TST, ultracentrifugation, SECMALAs can be used. And as we mentioned, it's a complex phenomenon, and there can be different nature of the peptide aggregates can exist in the peptide molecule. So the choice and selection of analytical method may vary from one peptide to another peptide. With this, I would like to invite Jawahar to conclude this session.
Thank you, Dinesh. So as a summary and conclusion, you have seen that peptide aggregation is really a complex and significant phenomenon. We have seen that our experience at aggregation will occur mainly during the downstream process steps, and it's influenced by various factors, intrinsic and extrinsic factors. So the dimension for process development of aggregating peptide and process control strategy for large-scale manufacturing focus on the complete understanding of the parameter that can influence the aggregation propensity, with the free axis, composition of the matrix, the mechanical stress, and the execution condition. So to end our presentation, we would like to thank our team at Polypeptide, the process development team, the analytical development team, and regulatory affairs team for their contributive input to better understand the aggregation phenomenon and to overcome all our challenges. And I would like also to thank you for participating to our webinar, and we will be very pleased to answer all your questions.